Hey, Baptiste.
How are you, Aaron?
Good to see you.
Good to see you too. Been a while.
Yeah. Do you want to give your slides a test?
Yeah, it was good.
Yeah, great.
Okay. Do you want me to unshare for the moment, or
Sh yes, please. So things in Ottawa are okay?
yeah, it's back to normal, I would say.
Mm.
What about you? How long ago did you go back to something looking like normal?
I'm still on a sabbatical, so I've been in
Oh,
Shanghai for a while.
okay. I didn't know.
Yeah.
So you're
Um,
streaming from there
yes.
now?
Yeah.
Okay, cool.
Yeah.
Okay. So do we start by me or?
Yeah, and
Okay.
I'll like say a brief introduction and then we can get started. How's Simon doing?
Simon is doing great as usual.
Good.
Very productive. We're just back from a meeting in Toronto with lots of
That was
fun.
one of my funnest visits is when I hung out with you guys.
Anyway, you should come back then.
Yeah.
We'll definitely find a way.
Yeah.
But we set the bar very high, so we have to find an even better place and
Yes.
venue for you.
Yeah, that was it was like a restaurant, but it felt like someone's sort of living room.
Yeah, someone's house. It was a really nice setup. I remember.
Mhm.
Check something. So, what are you doing from China? Are you expanding your expertise, doing some
Sure.
more?
Oh,
Yeah.
I mean, we're always expanding our expertise.
What specifically?
Um, Nothing specifically.
Okay. Okay.
Yeah, yeah, nothing specific. Hi, Zheng, yeah.
Hello.
Hi,
Hello, Eric.
hi, hi. Good to see you.
Hello.
Hi. Can you hear?
Wait.
We can hear you. Can you hear us? Can you hear me?
No, I cannot hear. I'm looking. Where is the button?
So,
Hmm. Sorry. Hi. Hey, sorry, Aaron. I'm looking for the button that can adjust my this video audio thing. So where is that?
Aaron.
Top left. Yes. Wait. Zoom. I use my headphone. Wait, I just can't see where I can click.
so
No,
like,
I can see Zoom meeting. I can see this
yeah.
thing, live YouTube, and there is no button for me to press.
So click. Do you see the menu on the top? It says Zoom.us, meeting, view, edit. So click on Zoom, the Zoom one, and then there's like,
Not
then there's
sure.
prep.
But you already are, right? What is Zheng looking for?
So she can't hear us.
Oh,
What?
I'm like,
you can hear me, but
we can hear you.
Yeah. That's weird.
Oh wow, great.
It must something happen with the computer, right? It's not.
I'm on
Zoom
the phone with. I'm on the phone
Yeah,
with her. That's how
yeah. I think it's computer problem. It's not a Zoom issue normally.
Maybe can you turn your is your volume too low? Is your volume low or muted on your computer?
Maybe just
I guess it's more important that we hear you than you hear us.
yeah. Your volume is down, seems.
Let me try again. So it's good that
It was, and yeah, maybe sometimes just restart computer. It works for me sometimes. Yeah.
Yeah. But do you see a Zoom?
Or exists a Zoom come back? Yeah.
Is there a Zoom menu on the top? Like, is there a menu for Zoom?
I click this connecting audience thing.
Okay.
Because I can see I'm speaking, then it's a response. It's a responses.
Maybe just come back. Just exist the Zoom will come back. Yeah, just works sometimes.
Okay. Do you want to try that? Close Zoom and then reconnect.
Yeah, come back. Exist and come back. Works for the most of the time.
How about that, Zheng? Do you want to log out and log back in, or quit your Zoom and come back?
Yeah, it'll work. Okay. It should work for most of computer problems.
Yeah.
Baptiste.
Okay. No problem.
How are you?
No problem. Oh, hey, Zheng.
Hey.
So all my friends are telling me they heard on the news that Shanghai is open now, but I'm telling them I didn't see that news because it's not.
It's only in news, not in the whatever. So,
I went
meta
to
world, not the
yeah,
real world.
I went to the
It's
gate.
a metaverse.
I went to the gate. I said, "Oh, I heard on the news we can come out," and the guards like, "Get back! It's not open yet."
It's happening in the metaverse.
Yeah.
Another Shanghai, not the.
Yeah. Yeah, I saw on the news people are going
In
out
a few weeks
shopping,
though, I
doing
thought,
stuff.
yeah.
Yeah.
But when's your flight? It's beginning of June.
Yeah, beginning of June.
Okay. A couple of weeks away.
Yeah. Hi, Jen.
Jane,
Hi, Aaron.
Hi.
Hello.
congratulations.
Yeah. So, well, they said that June 1st is when it's open, right? So then you can get to the airport.
I'll believe it when I'm in the air.
Okay.
So as long as the flight is still there, I think it should be fine, right? Sometimes the flight is canceled. The flight is still there. You should be able to get on it.
Yeah, but I might delay still. I'm like, if things are still tricky, I'm not gonna just leave my family behind.
Yeah.
Zheng,
Just wait a couple
can you
of
hear
weeks.
us?
Mm-hmm.
Zheng, can you hear us now? No?
Oh, yes, I can hear you. Yes, I can hear you. Finally.
Oh, yeah.
Finally
See,
it works.
the computer.
Great, yes. Good point.
It's
You're
always
quitting
works.
and
Come
entering
back.
again.
Yeah, and you can restart a computer. We can solve 99%.
Yeah, because my computer crashed once during the lockdown, so
Yeah.
I'm always worried
Yeah.
that they're gonna stop working again.
Oh, we can hear pretty well. That's funny.
But Zheng, did they tell you when you can go out? Because they told us at least another week here.
Yeah, for us it's beginning of June. Something says news, not says about anybody.
Yeah.
But the things for some not district Songjiang, they can indeed can drive out without any permission, without. So it's happening.
That's pretty far away though. Songjiang is not.
Yeah. I mean, so it means that at least it's happening somewhere in Shanghai.
Okay.
Yeah, anyway. All right, so get started.
Great. Okay. Hi everyone. Welcome to another Neuro Zoom edition. And looking forward to the talks today. And before we get started, just to announce the next week's speakers, we have Mian Zhao from Singapore, Duquesne, US, and we have Li Li from NYU Shanghai. She's not just NYU Shanghai; she's literally like my next door neighbor, living like right across the way. And just met her when I was walking my dog, and I said, "Oh, why don't you give a Neuro Zoom talk?" So we'll be cool to see what she talks about. And so tune in next week. Sign up. Let us know if you want to give a talk about your research. We're looking forward to having an exciting spring and summer talks. So tune in, check out the website, sign up for the YouTube channel as well. Okay, so now it's a great pleasure to introduce our first speaker, Baptiste Lacoste. He's an associate professor at University of Ottawa, and I knew of his research for a long time before I got to meet him, and it was great meeting him. He started out doing an undergraduate degree in France at University of Bordeaux, and then came to Canada, University of Montreal for a master's and PhD. He did a postdoc at the Montreal Neurological Institute in McGill, and then he did, where I really got to know him, he did a second postdoc with Chenghua Gu at Harvard Medical School. And here he started studying the interactions between the vascular system and the nervous system. And it's sort of been known for a long time that neural activity can affect blood flow. That's the whole basis for fMRI. But Baptiste solved a different problem in a very important paper during his postdoc with Zheng, where he found that neural activity also induces the rearrangement of vascular networks and changing of vascular structure. And this is important, very important implications for how sensory information can change the vasculature. This has launched him into his own laboratory at Ottawa, where he continues to study how neural activity and can trigger changes in vascular networks. So looking forward to hearing your latest, Baptiste.
Okay, do you hear me?
Yep.
Okay, awesome. Thank you so much for the invitation first and then kind introduction, and really happy to see you again. So today I'll actually present something which is very new and unpublished about the regulation of cerebral vascular growth by astrocytes. So let's first tell you this work, of course, has been done by talented people in my lab. Moses, postdoc in my lab, has done most of the work you'll see today in collaboration with an undergrad who has done most of the, you'll see the anatomy at the beginning, and Julie, who participated, and a lot of collaborators in funding. So astrocytes, lots of things are known about astrocytes, including something that you can see here. If you look at a staining, or in this case, LDH1L1 EGFP mice, we can see a lot of astrocyte cell bodies, but also those ghosts of highways, which are blood vessels, because astrocytes are actually covering blood vessels by their end feet. However, the way these interactions are set up during development is unknown, and actually the timing of when those interactions are established is still unknown to date. So before I dive into the meat, let's just put everybody on the same page. What are astrocytes? For those who don't know, they are specialized glial cells of the central nervous system. They are more numerous than neurons, and you actually find in the brain two types of astrocytes: the fibrous astrocytes, which are mostly found in the white matter, and the protoplasmic astrocytes, which are in the gray matter. Most of those we speak when we talk about neuro-glio-vascular interactions. But what do these astrocytes do? Again, I'm not going to summarize the whole field, but the main thing that we are interested in, for instance, are the fact they secrete pro-angiogenic molecules to regulate vascular growth. They extensively surround vessels with their end feet. They are thought to, so I'm going to be very cautious here, thought to, because it's not, and still a bit controversial, regulate maintenance of the blood-brain barrier, but they also serve as a bridge between neuronal activity and vascular control of vascular tone, mediating the effect of neuronal activity, as Aaron was mentioning earlier. But what is unknown is how clearly those connections are established, and this control of vascular growth in particular is established. And if you just look at the timing through which those two elements are developing, it's interesting to see that something which is already known, astrocytes develop and mature mostly postnatally, and it actually correlates well with angiogenesis in the brain and vascular maturation. However, and this is what I'm going to touch upon today on entirely unpublished data, is that the timing and the molecular factor which are regulating those physical and functional interactions is unknown. So the first thing we tested and we assessed to tackle this question is, first of all, looking at things over time. So we leverage expression of CD31, which is another endothelial marker, LDH1L1, which has been discovered by the lab of Ben Barres as a pan-astrocyte marker, but also GFAP, which is to me and my team not a good marker for astrocytes, and I'll show you why. And we also, of course, will show you some merge images and segmentations. So at P0 at birth, we can see that, something which I've published in the past and from Zheng lab, vascular networks are not very dense. But what we see here is astrocytes tend to look like immature cell, and so I'll show you a little bit more high magnification in a few slides, but they seem that they are climbing along the vascular networks to reach their final stage and the final positions. At P5, vascular density is slightly modulated, but astrocytes dramatically change morphology. And if you look at GFAP, it actually doesn't really label astrocytes at all. And you'll mainly find some radial glia and some places where astrocytes are expressing a little bit of GFAP. And the merge image is shown here. And to our surprise, this is only from P7, an age where vascular density started to increase, that we start to see actual physical contacts between astrocytes and vessels, which I'll show you are what we call endfeet. And again, GFAP doesn't really look like a bunch of astrocytes. And at P14, 2 weeks after birth, and P21, this is what looks like what most of the people working on astroglial vascular interactions are used to, which are those vascular ghosts. So let me show you a little bit what we quantified out of this. Is the two first graphs are basically what I already published, but it's always good to replicate your own work. Vascular density and branching patterns increase dramatically between P7 and P14 to reach basically almost maturity at P21. But if you look at astrocyte density, the number of cells per unit of tissue, then there is a peak at around P5, and then there is a global regular increase until about P14, which is completely finished at P21, about the contacts between astrocyte and vessels. So this is all quantified here, and just a little information here. The peak of astrocyte cell density was correlated with an increase in proliferation, which is actually already known. So if we look a little bit closer, we see that at P0, those astrocytes are not really formed in terms of morphology, but they seem to be quite associated with the vasculature in a different way than the endfeet, but rather using them as ladders. And then they detach, they mature, they get progressively a mature morphology, and then they form those classical endfeet. So we've quantified a bunch of things. This is just to show that the morphology get more complex over time. And again, around P14 and P21, this is where the astrocyte reach kind of their morphological maximal complexity, and their processes, which were all oriented towards the surface of the cortex, tend to spread out, and the angular entropy increases over time. But in terms of endfeet, I think I'd like, and I hope I'm going to convince you more if I show you electron microscopic images. At P0, you see astrocytes that are actually touching vessels, because we've seen they probably are migrating along the vasculature. But this is only from P7 that those endfeet are starting to form, which wasn't known to date. And we still have places where the vasculature is not covered. And at P14, well, this is pretty much what we are used to, with a thin layer of endfeet in green, in this case, covering the entire vascular wall. This is quantified here, just showing you that astrocyte coverage is increasing and reaching a maturity at around P14. The number of endfeet is refining and increasing again to reach a maximal around P21. And the total endfoot area decreases because those endfeet which are forming actually become smaller and smaller. So the next question was, well, it's pretty, those interactions now we know start around the first week of after birth, but is it genetically regulated? Can we find factors that actually control and regulate these processes? So recently we collaborated with the lab of Natalina Salmasso at Carleton University, and they published a database showing progressive expression patterns during postnatal development. And we looked at the database and found very interesting to, so first of all, focus on genes that are already known to regulate vascular development and remodeling, including HMGB1, which we confirm using our own spatial transcriptomic analysis, that HMGB1, and I'll dive into what this is doing and what is this gene, is very, very highly expressed at birth, much more than genes that are regularly known to participate in angiogenesis, such as VEGF-A. We confirmed this expression by Western blot, or this production of the protein by Western blot, very highly expressed at birth, decreasing rapidly, and we confirm this expression by astrocyte, but also by other cells. So validation of expression, this HMGB1 protein, which is a regulator of chromatin remodeling, is expressed by many cells, and its expression drops rapidly after P0, and it's expressed by glial cell, but only by astrocytes and by also neuronal cells. So you can see here that neurons express HMGB1, microglia don't, and endothelial cells don't, at least in terms of protein production or detectable amount of protein. But why is this interesting? Well, HMGB1 has only been shown to be involved in vascular repair after injury in a damaged adult brain. So it's very, very surprising, actually, to find it highly expressed at birth, when this gene, for instance, is known to repair vessels after stroke or to be involved in anti-inflammatory processes in the aging brain. So the question was, what is this gene doing at this stage of life? And well, the best way to test this question is to remove it from astrocytes. So we used a Cre-lock strategy where inducibly removed HMGB1 from astrocyte thanks to the Cre-ERT2 driven by the LH1N1 pan-astrocyte promoter. We treated early with tamoxifen and assessed at different time points. So we assessed at P14, 2 weeks after birth, where I've shown you things are pretty much formed, and P50, which should be considered as young adults. And we looked at vascular function and also behavior. We first confirmed that the Cre is indeed induced after tamoxifen treatment. The Cre is induced in astrocytes. And we also confirmed that we can selectively remove HMGB1 from astrocytes. So you see that expression or production of the protein is kept in surrounding cells, but exclusively removed from astrocytes. So what does this do? Well, it does not affect astrocyte density. It didn't affect how these cells are produced or formed. It did not affect, very surprisingly, vascular density and branching, so angiogenesis in the cerebral cortex. But when we looked at the proteins which are very characteristic of astrocyte networks and the way they interact with the vasculature, such as connexin 43, we were actually very surprised by the phenotype. So let me just first show you a normal brain at P14. The blue staining, immunostaining, is connexin 43, which is a very typically diffuse punctate staining. And on bigger vessels, you actually see those little zones or windows, which are single endfeet of astrocyte, because at the contacts between endfeet, connexin 43 is going to be highly expressed, and we can see those very beautiful pattern. But in the mutant, that was very surprising. Excuse me, like Siri is asking me a question. I have my watch. So in the mutants, we were very surprised to see a dramatic phenotype where the connexin 43 distribution was not that diffuse anymore, but also we were completely losing the vascular organization in little zones, and the astrocyte tended to sequester, to keep connexin 43 in their cell body rather than distributing it in their small processes, which is basically quantified here. But then if we are looking at those specific interactions, physical interactions between endfeet and small processes and vessels, well, let's look at EM because we have this tool. And let me show you on the left a normal neurovascular unit where in pink or red, you have the endothelium, a pericyte in yellow, and in green, the endfeet at P14. Well, in the mutant mouse, we observe dramatic phenotypes. And I'm saying phenotypes as because we not only found that the endfeet were fragmented, smaller, and disorganized, but also showing some places where the vessels were not covered anymore. But the most visually striking phenotype was actually the endothelial cells, showing vacuoles of macropinocytotic events. And I don't know if you can see those arms extended by the endothelium and closing on itself, which is an event very typical of macropinocytosis. And this is quantified here. So I'm not going to go into many details for reasons of time, but we see that the number of endfeet is increased, but those endfeet are smaller. And we actually see bigger numbers of macropinocytotic event in the endothelium. If we look at closer the endothelium, you are going to find many evidence of macropinocytosis, even those lasso type structures. And also surprising, for which I don't have any explanation yet, but we see those protrusions of the endothelium inside the astrocytic endfeet, which is quite surprising. And despite this highly disturbed endothelial structure, we didn't find evidence of blood-brain barrier impairment, where injecting a very, very small tracer, which is called cadaverine, we couldn't find it leaked out of the vessel as compared to a positive control situation where we actually did a wound in the cortex and you can see cells taking up the dye. Using EM, we also showed that tight junctions, which are those very specific junctions between endothelial cells of the brain, are not disturbed in this mutant. And we did a quick look at caveolé transcytosis, where with the numbers of caveolé vesicles were not actually changed, which are indirect indices of normal BBB function. Well, we modified the brain of these mice right after birth, and we looked at 2 weeks after. But what happened in the adult brain when you wait a little bit? Well, if you disturb glio-vascular interaction, it is very probable that we are going to disturb neurovascular function and neurovascular coupling in particular, which is a phenomenon mentioned by Aaron during his introduction, where when you stimulate neuronal activity, such as whisker stimulation, and look at neuronal activity induced blood flow, which can be done in the somatosensory cortex using less Doppler flowmetry, well, we actually find a slight yet significant phenotype where the amplitude of the blood flow increase to whisker stimulation or to neuronal activation was reduced, which is quantified here. However, the kinetics of these events were not affected. It was as fast as in the wild type mice or in the control mice, but not as strong. This was correlated with presence in the adult or maintenance in the adult of this connexin disturbed connexin phenotype, and not only maintained phenotype, but also aggravated, because when we quantify the size of those connexin patches, they actually looked bigger in the adult brain. And lastly, we started, again, this is unpublished and ongoing, but we started to look at behavior in these young adult mice, and we found one evidence of disturbed behavior using the marble burying test, where they actually buried less than the control, but they didn't look more anxious as assessed by the open field, or cognitive function didn't seem to be affected using the novel object recognition test. So this is still ongoing. We have a lot of work to do, but if I want to conclude, what I've shown you today is Astrocytes, which was unknown to date, initiate their vascular coverage during the first postnatal week, and that Astroglial HMGB1 appears to be required for maturation of this system. If we alter or remove HMGB1 early, we actually are going not only to disturb endfeet structure, but also endothelial ultrastructure, and which means that when you let the system mature with this defect, we actually see that phenotypes persist into adulthood, which was something interesting, and led us to think that, well, we are revealing another mechanism to regulate glio-vascular maturation. And the next question, of course, is going to be look for events in the aging brain of these mice, such as maybe strokes or different way that these mice age, such as neurodegenerative disorder. So I'm done. Thank you very much for your attention, and I'm very happy to take questions if there are any.
Awesome talk. Thanks so much, Baptiste. We're open for questions.
Hi, Baptiste.
Hey, sir. Guy, how are you?
How are you? Thanks for your great talks. Really nice. I'm glad I tuned in at 8 p.m. on a Monday after a long conference. So I have a quick question based. I really enjoyed the kind of part with the changes in connexin 43. And I was wondering whether that's, a symptom or, or is it causing the effects you're seeing, right? Is with the connexins and the network shuttling things from blood vessels to neurons and maybe back, right? Is the connexin 43 happening? Is it causing it or just a side effect?
It's an excellent question. It's actually hard to answer, because when you think about the fact HMGB1 is a regulator of chromatin remodeling, it actually could affect a lot of things in the astrocytes. I think you're right. Connexin 43 has been involved in angiogenesis, in vascular maturation, but I think that we kind of, and I'm telling this story honestly in the way we've done things, but I think it might be just a readout of disturbed endfeet remodeling. But you're right, we don't know yet exactly the causality, the cause to effect, and again, it could be HMGB1 also, the released portion of HMGB1 acting on connexin, which then induce disturbances in the system. But for now, without any more proofs, I would say this is a readout of disturbed glio-vascular structure.
Okay, thank you very much.
Anusha?
Whoops, sorry. Wasn't really ready to be asking questions, but hi, Baptiste.
Hi, Anusha, how are you?
Great talk. It was really nice to hear the latest. I also kind of have a question related to the connexins. I wonder if you looked at connexin 30 at all, and whether these changes in 43 are, like if there is any compensation happening for something else being lost, or if you plan on looking at like functional connectivity of astrocytes at all.
Absolutely.
Yeah.
Excellent question.
Kind of a follow-up on that is because of this glio-vascular interface being disrupted, I wonder if you've looked at any other things like glutamate transporters, or even like water channels, potassium channels that are important at that glio-vascular interface.
Yeah, so we plan on doing several things before getting this paper out in the wild. We are going to look at aquaporin 4. I would say related to parasites. We could look at connexins which are also specific to parasites. Yes, we are going to look at different proteins. And astrocyte connectivity, I've set up an experiment recently with my postdoc. So Moses is an expert in connexin 43. He's been doing a previous postdoc in Chris North's lab at UBC, and he has an assay where you can actually test the diffusion of a dye through connexin 43 in the cortex, and this is going to be done in the brain or mutant mice. So I'll definitely come back with more data. But thank you for your awesome question, Anusha. Nice to see you.
Andy?
Hey, Baptiste.
Hey, Andy. Long time.
Can you hear me okay? Hey, I'm cooking dinner on the side over here, so multitasking. So you showed these really interesting intravascular sort of protrusions that would go in, and the astrocytes sort of creating sort of strange sort of interfaces at where the blood cells might touch the endothelial wall. So that implies that maybe there's like a basal perfusion deficit, right? And I think, I'm not sure if you in the past you had showed me whether or not there was like a baseline blood flow difference, but I also wanted to mention that sometimes it's not just like global blood flow decrease, but there could be like little, like, occluded blood vessels here and there. And so Craig Brown has this really interesting like micro bead, like the dilated micro beads. When you inject that in there, and it shows you where all these little impaired vessels might be. And I wonder if you thought about trying that out. So
Andy, since we discussed during the weekend in Toronto, I've actually went back to the EM images we have, and you know what? I think you actually see, we didn't look on the low mag fluorescent images, but we actually see a lot of closed vessels, which are actually seem not perfused, and I think we are catching a lot of string vessels. So I'm actually going to go back to things that you've shown in your talk during the weekend, and we see a lot of evidence of vessels that are actually collapsed or not perfused. So yeah, that's an excellent point, Andy, and it pushed me to look further into the data.
Awesome. Great talk. Thank you.
Thank you, Andy.
David, please.
Oh, I think you're muted.
Hi. Sorry, I was muting myself. I've never really thought about this before. So HMGB1, is that acting intracellularly or extracellularly?
So there are two forms. There is both, actually. So one form regulates chromatin remodeling intracellularly in the nucleus, and there is a portion of it that can be secreted and act at distance. So it's definitely possible that the endothelial phenotype we see, if it is not a contact-dependent phenotype, might be a released portion of HMGB1. And we are setting up an experiment of a conditioned medium with a primary culture of astrocytes to check the proportion of released HMGB1 that would be required for, let's say, endothelial maturation. But you're right, there are two ways that HMGB1 can act, and we have not assessed.
Is there a way, can you do a rescue experiment in your glial knockout now with different portions of the molecule to distinguish these two things? Is that a feasible experiment?
That would be fantastic, probably requiring another year of work. But I think, I would say, fortunately, we found this gene which does a lot of cool things, but also a very complicated process where it's actually going to perturb a bunch of genes. So bringing it back, will it be sufficient to rescue the system, or
yeah, I think you could distinguish at least.
Yeah, at least this one. So you know what? Just to follow up on this question.
Yeah.
Yeah, just to follow up on this question, we can actually look at a system which already exists in the brain, which is the hippocampus. Recently, a paper was published in Cell Report showing that all the astrocytes connect blood vessels in the cortex, but not in the hippocampus. So we can find portion of vessels which are actually not contacted by vessel. Does this contact regulate endothelial growth? So that's one thing, maybe not dependent on HMGB1, but at least on contact. And the other thing I wanted to mention is if you look at the neurovascular unit or the endothelium of our mutant mice at P14, it actually looks like the endothelium at P5 in a normal brain. So it seems that absence of endfeet brings you the endothelium back to how it looked like or was at P5. And we are working on gene expressions data sets to look deeper at these questions on both cell types. Thank you for your question.
Thanks.
So Baptiste, I have a few questions about HMGB1. So it's also known that it shuttles from the nucleus to the cytoplasm, and I'm just wondering if, and that seems to be important for sort of cytokine release and the maybe the secreted form of it. But I was wondering if you just looked in the development of your system, if you see a change in localization of the protein.
So that's an excellent question. What we see with, let's say, if we just use immunohistochemistry like we did, either we don't detect what's in the cytoplasm, or we just see the nuclear one, but we just follow nuclear staining. Now the released portion could be tested using maybe mass spec, proteome, or metabolomics. But that's a great point. We don't have an answer for this, but we just see with immunohistochemistry the nuclear portion of HMGB1.
And also, so your genetic data is compelling. If you remove this gene function from astrocytes, you see an effect. But because of this possibility of it being secreted from neurons, for example, yes. Released from neurons, I just wonder if you've considered removing it from neurons and seeing if that doesn't have an effect.
No, so we considered, didn't do, but yeah, that's a great point. Yeah, I think we have the tools, so we could definitely check using and then MX13.
It seems like, and there are also some chemical inhibitors of HMG1.
Yeah.
P1. I wonder if those could be sort of useful for like you could just do IP injections. I mean, it's not as specific as your conditional knockout, but that might give you.
No, but we could try to treat a young mouse and see if it phenocopies what we see in the mutant.
Yeah. Okay.
Yeah, that's a great point, Aaron.
Cool. I've been reviewing papers on HMGB1 like crazy. I don't know why, but so that's why I've been learning a lot about.
Thank you.
Anyway,
awesome points.
Cool. Yeah. Awesome talk.
Thank you very much.
Yeah, great. All right, Zheng. Zheng is up next. You just have to unmute. Zheng.
Okay, thanks, Baptiste. Amazing talk. Now it's my great pleasure to introduce my colleague Zheng Ye, who is also a PI in ION. So Zheng was trained as a psychologist, but not a traditional psychologist. He uses all kinds of neuroscience tools to study psychological problems. So she has graduated from Zhejiang University, has bachelor degree in Zheng College. And after that, she went to Peking University, had studied psychology with Zhu Xiaoming, had PhD in psychology. After the PhD, she went to Europe, did postdoc in Germany and Cambridge. After 5 years postdoc, she went to Beijing to start her own lab in Institute of Psychology, also part of Chinese Academy of Sciences. In 2018, she joined ION. Today, she will talk about an interesting topic I heard a little bit before. Used a kind of interesting tool to study the sequential working memory. Apparently, she's studying interesting nuclear subatomic nucleus and the sequential working memory. Welcome, Zheng. We can see you now. Yeah, take it away.
Thank you for the introduction. So in the next 20 minutes, I'm gonna give this talk about subceramic nucleus and the sequential working memory. So first of all, why sequential working memory? So we all cook, especially during the lockdown time, and we have to. So, cooking is not only about adding the right ingredients, but also about adding them in the right order. And there are many activities, sequencing activities in our daily life like this, that is, the critical thing is to arrange the thoughts and actions in the right order. And it seems effortless for us, however, for patients with Parkinson's disease, it could be challenging, even though most Parkinson's patients do not have dementia, and they could have difficulty in non-motor sequencing tasks like this. So 30 years ago, a group of Greek psychologists and neurologists found that they let the patients to listen to the story about a sequence of 2 events. And because the language gave us the freedom to express these events in multiple order, they can use temporal connectives like after, that in which in after sentences, these two events were expressed in exactly the same order as they happen in the real world. So the linguistic order is consistent with this the real order, or in the before sentences, the order of these events were expressed. Well, the 2 events were expressed in the opposite order. So the story was told backwards. So the task is to understand the sentences and answer what happened first. And because there's multiple ways to express these 2 events, you can just separate them into 2 categories. One is this after style, that events what happens first always was expressed first, and the before one, which in which these 2 events were expressed always in the different order as what they happened in the real world. So this is the data. So if we look at those sentences in which these events were expressed exactly the same order as in the real order, and patients had no problem. They have relatively high accuracy of comprehension. However, if you look at those before sentences, that is the 2 events were expressed in the inconsistently as their like a real order of occurrence, then they will have almost the chance level or even lower. So we thought it's quite interesting, this phenomenon, and we look into the literature, and we found that it didn't matter whether those are the events or letters, numbers, words, pictures, or movements. So as long as patients were asked to rearrange those things, these items, they would have problem. So we thought, because this phenomenon happened quite early in their disease course, and it doesn't matter whether they are on medication or not. So we thought this maybe have something to do with the basal ganglia dysfunction. So if you remember the neuropathological stages of Parkinson's patients, there are 2 stages that we are particularly interested in. One is the damage of Lacoste aeroleus in stage 2, and one is the damage to substantia nigra in stage 3. And there are more reasons why we found that quite interesting, because first, Lacoste aeroleus is the primary source of noradrenaline neurons. It projects to the basal ganglia, and it has been associated with executive functions. And substantia nigra is better known, and it's the primary source of dopamine neurons in the brain. It projects to the striatum. It's not only responsible for motor symptoms in Parkinson's disease, but also dopamine is known to regulate working memory. So our first question is, does basal ganglia play a role in serial ordering? So, in particular, we are interested in, In particular, we're interested in serial ordering, but there are lots of other functions in sequential working memory you might be testing. So today, sequential working memory. So the first thing to do is we need a task which is friendly to patients, which can also be combined with, say, functional MRI, EEG, MEG, and deep brain stimulation. So the idea is we designed a digital ordering task. So the task is easy. The task is to remember always 4 digits in ascending order. In the half of these trials, the digits were already presented in ascending order, so there's no need to reorder them. We called it pure recall condition, and there would be a delay. After delay, there would be a probe, and it's asking, so whether, for example, in this case, 7 is the 4th digit in the target sequence. The correct answer to this case is yes. And in the other half of the trials, the numbers were randomly presented, and the participants always had to reorder them. So, and for the probe, it checks whether they did that. So if they did not do the reordering, and they should say yes, 7 is the 3rd digit of this sequence because 7 is presented as the 3rd digit of this sequence. However, if they do the reordering, then they should say no to this probe. And we first test them in a group of newly diagnosed and untreated patients with healthy controls. And the performance is exactly what we expected. So they performed relatively well, and there is a disease effect. So patients responded less accurately than healthy adults, and especially when they need to reorder the digits. So we combine this task with functional MRI. First of all, we want to look at the brain regions which are responding to the ordering processes. So although it seems quite effortless for us to just reorder these 4 digits, there are a group of distributed brain regions involved in these processes. So by contrasting random digits with ordered digits, we get the medial prefrontal cortex, dorsolateral prefrontal cortex, parietal cortex, and more importantly, the subcortical regions like subceramic nucleus, global pallidus, and the thalamus. And next, the more important question is, what is the disease effect on the ordering-related regional activity? So we have a look at the basal ganglia and the prefrontal cortex, and the good thing is there's only difference in the basal ganglia. So the overactivation of subceramic nucleus and the global pallidus, especially when patients had to reorder the digits, and there is no such change on the prefrontal cortex. So let's focusing on this STM. And then we found that there's a correlation between the ordering related STM activity and the ordering performance, but only in healthy adults, not in Parkinson's patients. So we get the first impression that this ordering function is lost in Parkinson's patients. So next step, we want to confirm that. So is STM actually contributing to serial ordering? And we want to do that with an intervention study. And we turn our attention to patients with deep brain stimulation, so we can manipulate. We can switch the DBS on and off and see whether the DBS on STM has any effect on their behavior, ordering behavior. And we can also go beyond the spatial resolution of functional MRI to look at the STM subregions, because STM usually STN has 3 subregions, like the motor, associative, and the limbic subregions. And they are linked with different parts of basal ganglia, and also different parts of the neocortex, and forming different loops, the motor, cognitive, and affective loops. And using the pre-operative and post-operative images, we can reconstruct the location of electrodes, and we can calculate the size of stimulation, and we can overlap these electrodes, the stimulation, with the atlas of the STN to find out which is the size of, say, motor subregions activated during DBS for each single patients. And just to avoid the potential bias introduced by one single task, we have 2 ordering tasks. The second ordering task is very similar to the one used by the Greek group 30 years ago. So it's the sentence here. And when patient read the sentence, they were then answer the question, what happened first or last? And they were answered by click the left button or the right button. So superficially, those 2 sentences, those 2 tasks are quite different. But what we want to see is what the two tasks in common. That is, by contrasting the random digits with the ordering, the ordered digits, we want to see serial ordering. And similar for sentence comprehension task, by contrasting the before sentences with after sentences, we also want to see the ordering, serial ordering thing. So it was surprising to see that when DBS is switched on, there is only negative effect on performance, because patients responded most slowly when DBS was switched on, regardless of the task, especially when they need to reorder events. This was so surprising because we go back to check the motor symptoms. It was significantly improved. Almost everyone, except one patient, everyone else got improved on motor symptoms. How could that be? The only possibility is, it could be that the motor function was improved, but the cognitive function was so negatively affected that the combination effect is just negative. So we look into the size of tissue activated in each subregion for each single patients. So although at the group level, we would say this normal distribution of this motor subregions, it doesn't matter whether the left or right, the motor subregions is always the mostly activated. And there are then associative subregion, limbic subregions, and so on. However, if we look into each single patients, yes, the majority of patients have bilateral stimulation on the motor subregions. However, there are patients who only got unilateral stimulation, or even no stimulation at all at the under this motor subregion. And it's similar for associative subregions, like more than half of the patients got stimulation on both sides, but there are patients who only got stimulation on the single side. And we double check the motor function, and it shows quite normally that the larger the stimulation on the motor side, then the better, the more the motor function would be improved. So the DBS actually worked, from the clinical view, the DBS works. However, the DBS effect on serial ordering is a combination of its effect on the motor side, the motor subregion, and the associative subregion, because, the more the associative subregion is activated, the performance is worse, and the motor subregion got the opposite effect, that the more the motor subregions was affected and the better the behavior performance is. So after confirming that the STN does contributes to serial ordering, want to ask one more question: what modulates STN? Locus coeruleus and or substantia nigra. So we go back to FMI study, but we need a new imaging technology, which is called neuromelanin sensitive MRI. And neuromelanin is a byproduct of synthesis of noradrenaline and dopamine neurons. So it exists in the noradrenaline neurons in locus coeruleus and the dopamine neurons in the substantia nigra. However, before the development of neuromelanin imaging, we can only see them in the dead brain. But because we want to measure cognition and we want to measure the integrity of locus coeruleus and substantia nigra in vivo. So we need this image method. And from the image method, you can see this high signal at locus coeruleus and the substantia nigra. So it would be much easier to quantify them. And here I show the real data. So the left one is the substantia nigra, the right one is locus coeruleus, and the top row is healthy adults, and the bottom one is patients. So you can see, can visually see that there's a big difference between them, the signals. So if we quantify them, the numbers would confirm what you just see visually, and there is a reduction of integrity of both substantia nigra and the locus coeruleus, regardless of the size, between patients and healthy controls. So then we want to combine this neuromelanin signals with our functional MI data. So first, we confirm, we replicate our previous finding that STN has this overactivation in patients compared to healthy adults. And there's something new that in addition, we found that there's underactivation of the striatum, the caudate nucleus. So we check the relationship between this STN and the caudate activity with the performance, ordering performance in this healthy adults and the Parkinson's patients. And we replicate the loss of STN function in Parkinson's patients, but we found something else. The caudate activity actually correlates with ordering performance in Parkinson's patients in this sample. So it looks like, so we got the impression that there is a switch of function between the STN to striatum in Parkinson's patients. And we further look whether the substantia nigra integrity can modulate, let's say, the ordering performance through the caudate activity, and the answer is yes. So the larger, the better the substantia nigra integrated is, the greater the caudate activity is, and the greater the caudate activity is, the better the ordering performance is. So in summary, this is basically what we found up to now, that in basal ganglia, STN and the caudate nucleus both contributed to serial ordering, and the substantia nigra may modulate caudate for the serial ordering. And because this project is still going on, and we also want to know what modulates STN, and in the end, to the end, the ultimate goal is to find out, to build a neurocognitive model of sequential working memory. This is what is roughly in our mind, but I'm gonna skip this because we have no time now. So that's all my talk.
Oh. Okay. I guess we could go a few minutes about the model. I'd love to see how it explained how the output of the STN go went to the prefrontal cortex. Yeah, we find for a couple of minutes. You can explain more about that.
Sure. So the model. So what we have in our mind is it should be a combination of prefrontal cortex and the basal ganglia, and the prefrontal cortex represents the sequence in our brain, and it's based on this competitive queuing model. What happened there is it will hold like these multiple items simultaneously. It will activate these multiple items in the same way, but the rank is represented by the strength of activation of each node. So if D is the first in the sequence, then it has the strongest activation in this prefrontal cortex. So when this one is being retrieved and generated output, then this node will be suppressed like this, and then the second strongest node, CB, will be the next output. So it works like this. And by adding the basal ganglia, we think, because this this model at the prefrontal cortex only explains how the sequence is encoded and retrieved, but we're considering whether how it could be updated. Let's say, well, this is the old the sequence DBAC. We want an updating, and the new sequence is ABCD. So how could this thing be rearranged to be ABCD? And we think we can maybe address this strength of activation of each nodes. But how can we do that, achieve that? The idea is maybe we can achieve that by from the contribution of basal ganglia, but I still don't know how. It's just roughly. We think this may be done by basal ganglia, but I don't know how.
Right. All right. Yeah. Go ahead.
Me, Richard?
Hello.
Yeah, Richard, please.
Hi. This is a very interesting presentation. Thank you so much. I wonder if you have considered studying the atypical Parkinson disorder, progressive supranuclear palsy. And the reason I ask is that although it is a very small region for PET, tau PET studies, especially with a 4R tracer, are showing that the STN is one of the most affected regions in PSP. However, I'm not aware of sequential working memory problems really having been studied much in that condition, or whether there are defects of the kind you have described in Parkinson's disease. And so I wonder if you have studied that or whether you would think about it.
Short answer is no, but I really, really want to. It's a great question and proposal.
Yeah.
Thank you.
Work with that tracer is going on in China, so it might be possible.
And to the listeners of this, to the audience of this talk, anyone who have this access to that PSP sample, and you can contact me for collaboration.
A quick question for the whether it's a for the DBS. So actually, when the on DBS on phase, the STN is activated or inhibited in patients.
Yeah, good question.
Right?
Well, it is still the exact mechanism of DBS is still on debate. But the idea, I mean, this is we say it's because there's the power is the larger power, but people said people said the beta the abnormal beta oscillation of STN should be suppressed when DVS was switched up. Yeah. I just took the easy terminology that the activation. Yeah. Sorry about misleading.
No. The things the kind of oscillation or activity in the STN to the sequence. That's something I'm thinking about.
The better way would be if I can recall these signals from STN. But the problem is I cannot record and stimulate at the same time. Yeah.
Maybe still need a lot of work.
Exactly. And lots of colleagues and students to do that.
Right. And network. Okay. And any more questions? Okay. No questions. I can, Aaron. I can put the section this morning. Baptiste and Zheng for talks this morning.
Thanks, everyone. Great job, John.
All right, thank you.
Thanks. Good to see you as well. Bye, everyone.
See you.